Hello, everyone, and welcome to this audio podcast interlude, this Quagmire interlude, this audio podcast in a Labonix interlude, this audio podcast as part of a Bason scroll, this audio podcast as part of a Storm Labonix production. In this audio podcast, audio podcast interlude, looking at sort of works in regards to sort of in around an excerpt, if you like, an excerpt as part of peer-reviewed and published works, peer-reviewed works, peer-reviewed and sort of published sort of retrospective works, and looking at works that links immunobiology, psychology, and also laboratory medicine in works looking at the laboratory investigations linking stress and the effect of stress on particular sort of cell types and central mechanisms. So in this interlude, in this audio podcast interlude, the following. So in literature, in vivo studies or investigations using interleukin 15, interleukin 2, interleukin 15 receptor alpha, stress cellular adhesion molecules, and natural killer cells and NK cells should be investigated with chemokine receptors such as CCR5, CCR7, and CXCR3 in a T cell culture system to see if this affects responses in patients undergoing various forms of renal replacement therapy. Utilizing peripheral blood mononuclear cells, PBMCs, would be advantageous. Again, utilizing sort of chemotaxis assays with flow cytometry would allow or aid to give sort of graphical data, linking specific endocrines such as cortisol, adrenocorticotropic hormone or ACTH, and glucocorticoid hormone may prove advantageous in investigating stress on cell types accordingly. Stress and sort of coping, for example, may have specific effects with regard to T cells migration, T cells being those cells again that prompt or elicit sort of a response for example red cells or red blood cells to react and various other cells to act to either favor physiological conditions at homeostasis, or to impact physiology in such a way that cause the host of those cells to sort of become sort of impaired in some way. So stress and coping may have specific effects with regards to T cell migration in recipients or patients undergoing renal replacement therapy, and instigating migration investigation is one of the most useful ways to look at T cell migration. Hormones that are generated endogenously through the endocrine system and by stress or in reaction to strain, by that are in reaction to strain and also where hormones are also recognized in modifying the pathway of T cell trafficking and augment consequent cell activity. Laboratory investigations could be replicated to investigate stress, for example, stress positive and stress aligned to positive and negative strategies in order to identify how particular immune cells are influenced in amongst those suffering with CKD and or on forms of renal replacement therapies. Understanding the complex interactions of stress still remain a challenge in the 21st century. That draws this audio podcast to a close. This audio podcast as part of a Quagmire interlude. This audio podcast as part of a Labonix interlude. This audio podcast as part of a Bason scroll. This audio podcast as part of a Storm Labonix production.